There are two categories that organisms fall into, prokaryotic or eukaryotic. The main distinction between them is that eukaryotes have cells with one or many nuclei where they store their genetic material. Eukaryotic cells also have membrane-bounded and non-membrane-bounded organelles, which are specialized structures with specific functions within the cell. In contrast, the cells of prokaryotes don't have nuclei at all, and they only have organelles without a membrane. But regardless of whether you are a prokaryotic or eukaryotic organisms, we all have ribosomes, and this makes sense since they are organelles without membranes. Ribosomes can be thought of as the protein factory of our cells. What they actually do is read and follow the instructions from the DNA of organisms to form long polypeptide chains. Once the polypeptide chains are complete, it is disconnected from the ribosome and folded into its final 3D conformational shape to become a protein. While ribosomes simply float freely in the cytoplasm of prokaryotes, in eukaryotes, ribosomes are also located on the rough endoplasmic reticulum, this long, flat membrane connected to the nucleus. Here is where we come in. Remember that our genetic material, DNA, is where we store instructions for making proteins. But as eukaryotic organisms, how do we get those instructions from the nucleus to the RER, where our ribosomes are? Well, the most simple answer may be to simply transport that DNA through these convenient holes in the nucleus membranes to ribosomes. But that is actually a really bad idea. Here's the thing, DNA is easily damaged. That's why it is safely stored in the nucleus. If we took DNA out of its storage whenever we need it, which is quite often, we risk damaging it. To solve this problem, we instead make a copy of the segment of DNA we need, and this copy is called RNA. RNA and DNA are basically the same except for a few key differences. 1. DNA is composed of the 5-carbon sugar deoxyribose, while RNA has the 5-carbon sugar ribose. 2. RNA is temporary and can be disassembled and reused after a protein is made. This way, we can preserve the unique sequences of DNA and use them when needed without the risk of damage. I'll mention the third difference in a little bit. Now we can finally get to the topic of today's video, protein synthesis. There are two parts to this process, transcription and translation. During transcription, an RNA copy is made from DNA, and during translation, that RNA is read by ribosomes and that protein is made. For the sake of time, I will go over transcription right now, and in part 2, I'll discuss translation. During transcription, a variety of proteins called enzymes are used to carry out the reactions of each step. First, the enzyme helicase is used to split DNA into two strands, the coding and non-coding strand. The coding strand is a strand of DNA that carries the information for the particular protein you need, and this is distinctly different from the non-coding strand, which is the exact opposite of the coding strand due to the complementary base pairing rule, and therefore does not have the info you need. After helicase, enzyme topoisomerase is used to keep the DNA strands from tangling. Then a segment of RNA called a primer is attached to DNA, marking the starting place of transcription. Finally, the enzyme RNA polymerase is used to bind to DNA where the primer is at and begin transcription. Note here that without a primer, transcription cannot begin because you wouldn't be able to attach RNA polymerase. What RNA polymerase actually does to create an RNA copy of DNA is to read the base sequence of the coding strand and use the rule of complementary base pairing, where thymine is paired with adenine and guanine with cytosine. RNA polymerase will grab the appropriate base and add it to the growing RNA strand. And it is here that we finally come across the third key difference between RNA and DNA. RNA uses the base uracil in the place of thymine in DNA. So let's say the next base read by RNA polymerase is adenine. Typically, you would add thymine to the chain. However, because this is a piece of RNA, you will use uracil, and this is how uracil replaces thymine. Okay, RNA polymerase is busy doing its job, and now it's come across the terminary sequence. After the terminator sequence is transcribed, the process ends, and RNA is detached from the DNA sequence. RNA in this form is referred to as pre-messenger RNA. It is called pre-mRNA because it is the unmodified version of the messenger RNA, the strand that will eventually be messaged out from the nucleus to the ribosome. But first, pre-mRNA needs to mature, and how does it do so? Well, you'll have to wait to see next time. Thank you.